In this week's video, we're going to talk about the senses. The first sense we're going to talk about is taste. The receptors for taste are located mainly on the dorsal surface of the tongue within taste buds, which are housed in the lingual papillae. Each taste bud contains 50 to 150 receptor cells, also called taste or gustatory cells. These are specialized epithelial cells that synapse with neurons and can excite their associated neuron when they interact with the taste stimulus, generating action potentials. The 5 basic taste modalities are salt, sweet, bitter, sour, and umami. Specific tastants like sodium chloride, sucrose, hydrochloric acid, quinine, and monosodium glutamate efficiently interact with receptor cells. There is a unique receptor for each taste modality, though other molecules can interact with them. The apical end of the receptor cell responds to the tastant, while the basal end transmits the signals to the brain. For a substance to be tasted, it must first dissolve in saliva. Receptor cells for salt or sour substances contain membrane channels that allow the salty ion or sour ion to pass through, causing depolarization. This depolarization opens voltage-gated calcium channels, and calcium diffuses into the cell, triggering exocytosis of a neurotransmitter from the basal membrane. Sweet, umami, and bitter sensations use G-protein coupled receptors. These substances bind as ligands to the GPCR, triggering a second messenger system that opens calcium channels on the cell surface or the smooth endoplasmic reticulum. The influx of calcium facilitates the release of neurotransmitter, which then elicits an action potential in the associated neuron. Taste perception allows the brain to distinguish sensations based on the perception of action potentials from different receptor types. Input from the sense of smell and input from other sensory receptors in the mouth, like temperature, texture, and pain. The perception of taste is processed in the primary sensory cortex of the brain, receiving action potentials via the facial nerve, glossopharyngeal nerve, and vagus nerve, which converge on the solitary nucleus of the medulla oblongata. The next sense we're going to cover is smell. The sense of smell, or olfaction, is our other chemical sense. Unlike taste cells, olfactory receptor cells are neurons. These neurons are located within the olfactory epithelium in the roof of the nasal cavity. Chemical odorants in the air must dissolve in a thin, water-based mucus layer lining the olfactory epithelium to be smelled. Dissolved odorants bind to receptors on the receptor cilia of the olfactory neuron. Olfactory receptors are GPCRs that activate the enzyme adenylate cyclase, which produces CAMP. CAMP then opens a cation channel, allowing sodium and calcium to enter the cell, depolarizing the membrane and triggering an action potential. This depolarization can also activate voltage-gated chloride channels, causing further depolarization as chloride leaves the cell. Olfactory sensory neurons synapse with neurons in the olfactory bulb that extend to the olfactory cortex. Signals pass to the temporal lobe for odor perception or to areas in the frontal lobe associated with the limbic system, generating emotions related to the smell. Olfaction is unique among the major senses in that it does not pass through the thalamus. The next sense we're going to cover is sight. To see an object clearly, light reflected off the object must be focused on the retina. The eye accomplishes this by bending or refracting light. The eye has 2 convex structures for bending light, the cornea and the lens. Their densities are different from air and the aqueous and vitreous humors within the eye. The cornea is the clear window on the anterior surface of the eye and is the major refractor of light entering the eye. Its transparency is due to the arrangement of collagen and proteoglycan fibers. However, the cornea's refractive ability can't be adjusted. The lens is located behind the iris and pupil. It is adjustable due to its elasticity and the actions of the ciliary muscle. The lens divides the eye into anterior and posterior cavities. The ciliary body is a part of the middle layer of the eye, and it is continuous with the choroid. It is composed of the ring of smooth muscle, the ciliary muscle, and the ciliary processes. The ciliary muscle is a sphincter-like muscle. The ciliary muscle is attached to the lens capsule via the suspensory ligaments. The ciliary muscle is responsible for adjusting the thickness of the lens. The closeness of an object affects how much the light diverges, requiring different amounts of refraction to focus properly. As the lens becomes thicker, its focal point shortens, bending the light more sharply. As it flattens, its focal point lengthens, bending the light less. The elasticity of the lens and the action of the ciliary muscles on the suspensory ligaments allows this adjustment. Photoreceptors and phototransduction. The retina is the structure in the eye responsible for converting light waves into action potentials. The neural layer of the retina contains two types of photoreceptor cells, the rods and the cones. Rods are responsible for black and white vision and night vision. They are very sensitive to light and can respond to a single photon. There are about 120 million rods per eye. Rods are part of convergent circuits, meaning signals from multiple rods converge on a single bipolar and ganglion cells, allowing summation of signals for vision in dim light. However, this convergence results in less accurate vision. Rods contain the visual pigment rhodopsin, which is most efficiently activated by light of green wavelength. During the day, in bright light, rods become inactivated through a process called bleaching. Cones are responsible for color vision and sharp vision. There are about 6 million cones per eye. Whatever that is. Okay. Cones are less sensitive than rods and require more light, making them effective in full daylight. There is very little convergence in cone circuitry, allowing for very sharp vision, especially in the fovea centralis, where only cones are found. Humans have 3 types of cones that respond to red, green, or blue wavelengths, and mixing input from these allows perception of about 1 million different hues. Cone visual pigments are similar to rhodopsin, but respond to different wavelengths. Cones recover from bleaching more quickly than rods do. The process of converting light into action potentials is called phototransduction. In rods, this begins with the visual pigment rhodopsin, composed of the protein opsin and a retinol and vitamin A derivative. In the dark, retinol has an 11-cis shape and fits into a binding site on opsin. When light is absorbed, the energy changes retinol shape to all trans, which detaches from opsin, causing bleaching. The activated opsin stimulates the alpha subunit of a GPCR called transducin. Activated transducin activates the enzyme phosphodiesterase, which breaks down cyclic GMP. Photoreceptors are unique because they release neurotransmitter when they are not being stimulated. In the dark, cGMP is bound to cGMP-gated cation channels, allowing sodium and calcium to diffuse into the cell, maintaining a depolarized state. This depolarization opens voltage-gated calcium channels, increasing calcium influx and triggering glutamate release by exocytosis. When light activates the photoreceptor, the breakdown of cGMP causes cGMP-gated cation channels to close the membrane, and the membrane hyperpolarizes. This hyperpolarization closes the voltage-gated calcium channels, stopping glutamate release. Thus, light inhibits glutamate release from photoreceptors. The removal of this inhibitory signal allows associated bipolar neurons to fire action potentials, which are then sent to the brain. Retinol must be converted back into the 11-cis state in the pigment epithelium to bind opsin again and reactivate the rod cell. When we talk about visual receptive fields, the ability to produce sharp vision and distinguish between various contrasts and edges is a property of receptive fields. Receptive fields are formed by the cooperation of photoreceptors, bipolar neurons, and ganglion cells. Receptive fields of photoreceptors are circular, with photoreceptors located in a central area and others making up peripheral edges known as center and the surround. Photoreceptors release neurotransmitter in the dark. Bipolar neurons associated with the center of a receptive field may be inhibited by this glutamate. Horizontal cells connected to the surrounding photoreceptors also influence the bipolar cell activity. If light strikes only the center photoreceptor, it reduces glutamate release, stimulating the bipolar cell. The surrounding photoreceptors remain in the dark, releasing glutamate via horizontal cells, which might enhance the effect of the center light. Conversely, if light strikes only the surround, the center remains in the dark and releases glutamate, while the surrounding photoreceptors reduce glutamate release, affecting the bipolar cell via horizontal cells in a way that results in less stimulation. The net effect of light on the center and surround determines the frequency of action potentials sent to the brain via the ganglion cell, contributing to the perception of contrast and edges. The last sense we're going to discuss is hearing. The ear is a sensory organ designed to capture, transmit, and translate sound waves into action potentials. Sound waves are pressure waves with alternating regions of compressed and rarefied air. What we perceive as sound is translation of frequency and amplitude of those waves. The first part is your external ear. The external ear consists of the auricle or the pinna and the external auditory canal. The auricle captures sound waves and channels them into the auditory canal. The auditory canal conducts the sound waves to the tympanic membrane. In the middle ear, the tympanic membrane is bound between the external and the middle ear. Its primary function is to vibrate in response to sound waves. The middle ear is an air-filled cavity containing 3 auditory ossicles, the malleus, incus, and stapes. These bones form a bridge between the tympanic membrane and the oval window of the inner ear. The malleus sits on the tympanic membrane, the incus is between the malleus and the stapes connects to the oval window. The middle ear transfers vibrations from the large tympanic membrane to the smaller oval window that amplifies the sound waves. This amplification is necessary because transferring vibrations from air to the fluid-filled inner ear requires more energy. The middle ear is connected to the pharynx by the Eustachian tube, which equilibrates pressure. In the inner ear, we have structures for hearing and balance embedded in the temporal bone. The structure involved in hearing is the cochlea, a spiral-shaped structure containing 3 fluid-filled chambers, the scala vestibuli, the scala tympani, and the cochlear duct. The scala vestibuli and the scala tympani are filled with perilymph, while the cochlear duct is filled with endolymph. The endolymph has a high potassium concentration. The oval window communicates with the scala vestibuli. When the stapes vibrates in the oval window, it causes vibrations in the fluid of the scala vestibuli. These vibrations travel to the helicochrema, where the scala vestibuli becomes the scala tympani, ending at the round window, which acts as a pressure release valve. Within the cochlear duct is the organ of Corti, or the spiral organ, which converts mechanical vibrations into electrical action potentials. It sits on the basilar membrane, which separates the cochlear duct from the scala tympani. The basilar membrane detects sound waves of different frequencies because it varies in width and stiffness along its length. Segments near the oval window are narrow and stiff. They vibrate at high frequencies, while segments near the helicochrema are wider and more limber, and they vibrate at low frequencies. This phenomenon is called resonance. Located on the basilar membrane are hair cells, which are the receptor cells for hearing. They have stereocilia on their apical surface. The longest stereocilia on the outer hair cells are embedded into the tectoral membrane, which extends over the hair cells. When a fluid wave causes the basilar membrane to vibrate, it bends the stereocilia of the hair cells. Bending of the stereocilia towards the longest stereocilium causes mechanically gated cation channels to open. In hair cells, potassium ions are used to depolarize the cell, which is unusual but possible due to the high concentration of potassium in the endolymph and the endocochlear potential, around positive 80 millivolts in the endolymph relative to the perilymph. The bending of the stereocilia opens potassium channels, causing potassium to enter the hair cell from the endolymph. This influx of positive charges depolarizes the hair cell. Depolarization opens voltage-gated calcium channels, leading to the calcium entry. The influx of calcium triggers the fusion of synaptic vesicles and the release of the neurotransmitter glutamate. Glutamate induces an excitatory postsynaptic potential in the associated neuron. Axons of these neurons from the cochlear nerve, which transmits actions potentials to the auditory cortex of the brain. At rest, about 10% of potassium channels are open, resulting in a low frequency of action potentials. Bending of the stereocilia further increase channel opening and action potential frequency, while bending in opposite direction closes channels and decreases frequency. The inner hair cells are primarily responsible for releasing the neurotransmitter that generates action potentials in the cochlear nerve, while outer hair cells amplify that movement. The pitch of a sound is determined by the region of the auditory cortex that receives input, as each region of the cochlea is hardwired to a specific region of the brain. The loudness of the sound is determined by the number of action potentials that reach the brain, which is a function of the amplitude of the sound wave. Higher amplitude waves cause more vigorous vibration and greater depolarization, leading to a higher frequency of action potentials. The brain can also fine-tune hearing using efferent neurons that synapse on outer hair cells and some afferent axons, suppressing hair cell responsiveness to filter out unwanted sounds.